Good afternoon to all of you. Sorry for some delay in the start of the program. So hope you will be able to be handy up to 1:30. Welcome to all of you for this important training. So today overall India has to reach about 1.6 million population by 2047. That is when we celebrate most of the Chinese will be in the helm of affairs at that time. We need to fill the India population. For that, we need to increase the food grain production by 1.64, fruit production by 1.96, similarly vegetable production by 2.13. At the same time, we have to also reduce the post-harvest losses. We have to increase the efficiency of the inputs. Two major inputs that are used in agriculture. One is the water and another one is the these two we have to increase. So all these require application of strategic science in agriculture. Then only we can achieve all this. So as we are fortunate that there are several techniques that in the scientific area of science that are available to us to address these stressing national issues and issues of climate change. So one of the most important as a crop improvement process, your aim is always to increase the genetic gain. So how we can increase the genetic gain? Either you have to increase the selection intensity or the selection accuracy or the genetic variation, or you have to reduce the generation time, and also you have to reduce the depression or the effect due to the various alleles that accumulate due to the breeding process. So genome editing is not such important tool. It can increase genetic variation. That is, whatever variation you want to create, you can create. The entire breeding program depends upon variation. Variation in the population, this variation can be easily created. So that is one important aspect that the genome editing can access. At the same time, it can reduce the generation time. That is, by using conventional breeding, you could take about 6 to 7 generations of breeding work. This can be achieved within 2 generations. At the same time, the imprinting depression or the alleles that accumulate due to their breeding procedure because of their linkage and other aspect can be overcome because you are editing specifically the target gene, so there is no effect of that linkage tract or accumulation of the LTDs. So overall, this genome editing is very important. The next generation to increase genetic gain, and thereby you can create plant at your will so that all the targets for 2050 and beyond can be achieved. So before going into the genome editing, which is a powerful mutation tool, we should know the power of the mutation. That is evident from these two pictures. I think most of the students will be able to recognize. One is the normal gorlus standing with this semi-dwarf wheat varieties, and because of the editing, this mutation in what gene, which is the delta protein coding, which is also known as RST. So you can see the power of mutation, and similarly on this side you can see the rice plant. The tall rice plant can be convert it into rice plant IRI is created. So this is again a single mutation in the GA20 oxidase that led to reduction in the GA20. So you can see that these two mutants, if you say that they alleviated the poverty and destruction of human race on the earth, that is not an exaggeration, such as the impact of mutation. So these are spontaneous mutations, and you get very rarely these mutations. But genome editing offers you such a strong tool. You can create any mutation that can bring changes in the crops and the lives of humans. There are several genome editing techniques earlier we had, which include based on the protein DNA interaction, which includes the meganuclease or the zinc finger nuclease or the, they all three rely on the protein DNA interaction. You know that protein DNA interaction activate a specific sequence in the protein, and it is very difficult to engineer the protein to target it to specific changes which need to be edited. So all this editing process is because of the DNA damage that is caused by the nucleases associated to the genome. So you have to target these proteins to a specific process, which is not possible to engineer by protein engineering and targeting these proteins to the specific sequence. This professors Jennifer Dornan, Emmanuel Charpentier, and they came up with the novel technique of RNA-guided genome editing. So now your RNA is going to guide the molecular scissors that is going to create DNA damage, which can lead to mutation. So this was done by the Jennifer Dornan and Emmanuel Charpentier. They used RNA as a guide to take the nucleus to a specific location. This is a history, and everybody might have recently I had an opportunity to visit Jennifer Dornan's lab, and we are making an effort to sign an MOU with ICR and with Jennifer Dornan's innovative genomic institute, so that we can train all our young scientists and students there in the Jennifer Dornan's Institute of Innovative Genomic Institute. So to start with the genome editing by using the RNA-guided genome editing, which is also called as the CRISPR-Cas genome editing. Although you will be listening everything in detail, just I want to introduce this topic. So it is to use two reagents. One is the guide RNA, which takes the molecular scissors to the specific location in the genome. So these are the two reagents, and these two reagents can be delivered into the cell. That is, you have to deliver this into the cell to edit to get the DNA damage, which can be prepared to produce. So this can be delivered as DNA. That is, we make a gene construct by using either a agrobacteria-mediated genetic transcription or particle bombardment or PG-mediated transcription of chloroplast. We can deliver this reagent into the cell. Where this can be transcribed to produce guide RNA and the Cas9. These targets can target a specific location on the genome to make this edit. Or alternatively, we can deliver it as RNA plus protein. That is, you need the guide RNA and its scaffold as RNA, and the Cas9 protein. You can mix these two and you can directly deliver into the cell. The plant cells have a major limitation of delivering the protein RNA complex, or what we call it as the RNA complex. So for this, we have to use only the protoplast-mediated transformation. So protoplast-mediated transformation has other limitations such as regeneration, and it is not easy for all the crops. So the common method of delivery is agrobacteria or particle compartment-mediated genetic transformation. So these reagents, you deliver these reagents, you make a gene construct and you deliver it as the DNA. So for this, you have this transcriptional unit in your gene construct. One transcriptional unit transcribes the guide RNA, plus the scaffold RNA. This is the guide RNA, which is about 20 nucleotides, and you have a scaffold RNA which helps in binding of the Cas9. And the guide RNA takes the complex to a specific location. So this guide RNA is contributed to the specific location of your interest. So this is just like a primer with what you design for PCR. So just design a primer which can bind to a specific location without targeting multiple locations. So this is cloned in front of RNA polymerase 3 promoter which can produce RNA. And the Cas9 is cloned. This is Streptococcus pyogenes. So this is Cas9. So this is from the bacterial source. So you are going to use nuclear localization sequences to target it to the nucleus. And this modified Cas9 is cloned in front of a constitutive promoter. It is a promoter which is generally used in dicards. Dicards are ubiquitin promoter which is used in the So this through transcriptional units are in a simple construct. You need to clone only your guide RNA. Rest everything will be there under the vector of your interest. Transform this into the cell. Once it goes into the cell, it produces RNA and it produces Cas9. And as shown here, this RNA Cas9 complex, it goes and binds to the target. So this when you design the guide RNA, you see that there is a protopressor activation which is specific to this Cas9. So in case for Cas9, it is depending upon the nucleus that you use. This protopressor activation molecule is different. For Cas12, it is different. For Cas9, it is different, and various modified versions will also have different the PAM sequence. So it will go and bind, and then once it binds, this Cas9 enzyme has two cut sites on the DNA. So it makes a perfect blended cut of the DNA wherever the target is there. It just exactly 3' upstream to the PAM site. You know where it is going to exactly cut. So to a large extent, you can predict where the mutation is going to occur. So once the mutation happens, that is the double-stranded break happens, then rest is taken care of by the cell. So once this double-stranded break is repaired by the non-homologous enzyme, it is a natural process of the cell, and it repairs the double-stranded break by its natural mechanism. Through this process, you will get an insertion or a deletion. Normally, insertion is one-base insertion. You get most frequently or majority of the cases, or you get a deletion of 1, 2, 3, or large deletion also you get sometimes, depending upon how the microhomology is there that repair. So this is kind of mutation that was created without help and repair is known as site-directed nucleus mutation. So when you discuss about regulation, you know what is SDN1 and SDN2. In other case, to guide the cell, how the repair we can also guide, because the SDN1 is a random process. We can get insertion, deletion, or any kind of variation. You can also guide the cell by providing a template, so that the cell can just copy that template on the double-stranded cleavage site. So you get exactly whatever the mutation you desire. That is, you can alter 1 or 2 bases in the original template. That is the homologous, which is similar to the native gene, except for 1 or 2 amino acid mutations that you introduce. In the nucleotides, you change across different amino acid. So that exactly will be copied, and you get only 1 or 2 nucleotide change as you desire. That kind of mutants of the SDN3 mutants, where the express that we use is a foreign gene. It has homology arms, that is, these are similar to that of the native DNA, and in between you can use any gene of your interest. So this one is again by homology dependent mechanism. It can be inserted, copied into the base where the double-stranded cleavage is there. So this can help integrating the foreign gene at specific location. That is, you have a strong promoter, you want to put only the foreign protein coding gene in front of the promoter. It is possible. And also you can, when you generate transgenic plant, you have to create a large number of event to get the event of. So in this case, if you know where to integrate with the gene, you will get a good expression. So you can specifically target the gene into that place. This will also help in avoiding the integration of foreign gene into the essential gene. If you remember the golden rice, the first golden rice which came for India for integration into the mega varieties, everything was done. Finally, it was found that it was integrated with the gene which is involved in the oxygen transport. Because of that, it could not be commercialized. So these kind of issues will not arise if you use genome editing even to develop a transgenic plant. So besides this SDN1 and SDN2, there are basic systems where you do not create a double stranded break in the DNA. You create only a single strand to the Cas9 modified Cas9 DNA deaminase. It can be either C or A can be deaminated so that it can create C2G or A2G substitution. Thereby you can create only specific gene without creating another double strand. So this is again no template is guided. This one is also exempted from the regulation. And the recent one is the prime editing where you can create all kind of mutation. There is no limitation either. You cannot create small deletion, small addition or small modification. So in this only the guide RNA is a little bit different. And here you can see the guide RNA which you normally use. The guide RNA is there. At the end of the 3' of this guide RNA you have editing template, which is that's why this is called prime editing guide RNA. So in this you have the sequence which is similar to this region. In the editing region, it's some few bases change. So this can be small, some revolution or addition or deletion of bases. Everything can be possible or modification of bases. And the Cas9 is again it is a case. It will not create a double stranded link. One of the base site is mutated. And to this Cas9 you have reverse transcriptase. So this complex once it makes a double stranded cleavage, the RNA binds to the cleavage site and then reverse transcription can create a new DNA strand, and then during the repair process the new sequence is integrated. It is template guided, but here the template is not DNA, which is very difficult to deliver into the cell, which some of your future lecture will listen from some of the other experts. So this is in the different approach you can use for editing. So normally when we talk of this one, these are considered as non-GM cross because in case of the transgene that is integrated into the plant that gives the trait. Finally the plant which is homozygous for the transgene and that is permanent and that gives the trait. Whereas in case of genome edited cross, you segregate the transgene which is integrated in one place on the genome, the target site is somewhere else or it can be nearby. So you separate the transgene and mutation to self-limiting segregation, so you get the which is free of any. So the mutation is taking place in the gene, it's native place, and there is no foreign gene is involved. That's why these mutations are not detectable whether it is created through genome editing process or it is through any of the induced mutations. So that's why these are primarily exempted from the biosafety regulation. So when we talk of genome editing, we always think that only many students think that we create only few variations. That is, you can just create knockout or you modify one or two bases. So that is not true. So in gene, how many elements are there? Functional elements can any student can say. If you tell gene, so what are there in the gene? What are the functional elements? Promoter, we have several elements are there in the promoter, and everything is summable for each of these elements. When you edit, you get different effect. That is not the simple mutation. So, for example, in promoter, you can have different cis elements. Each helps the expression of the gene at specific time, or it helps the infection of gene at specific time, and the other time it is in the normal level of expression. So it can be a pathogen inducing elements it will be there, as in case of the sweet trees, those who studied some this bacterial blight disease in rice. That remember that the zoo induces the sweet genes through cis element which are present in the promoter of the sweet gene. And if you modify that cis element, that is even when the bacterial effector is coming, it cannot bind to that specific place and induce the expression. So if you edit any transcription factor binding site, that transcription factor will not be able to bind. So there will be no expression. Alternatively, you can induce one or two nucleotide change because the cis elements are very small sequences. You can modify one or two nucleotide that are there in the promoter, so you can create new cis elements. That is, you can make a promoter which is indivisible or indivisible by any kind of stress, or you can create mutation to modify the expression in the different developmental stages. So all kind of cis element editing is possible. Next, the element in the gene is the 5' UTR. 5' UTR plays very important role in the expression of the gene, as the amount of protein produced also depends on the 5' UTR, whether it has the 5' UOR, that is the upstream ORFs or small ORFs that are present before the normal ATG codon. These codes for a few amino acids, maybe 7, 8 amino acids. So when ribosome starts searching its start codon, it will start its translation from the URL, and it will fall off after the URL. So a small protein is produced, and normal translation will not start from the ATGR to less extent. So if you want to increase the amount of protein without altering the expression of the gene, that is possible by editing the upstream ORF. Then there are a number of opportunities to edit the exon and intron to create all kind of modification. You can modify protein activity, as in case of the herbicide tolerance. You keep the enzyme activity same, but its binding to the herbicide site can be edited so that the protein retains its function, but it will be more active, or it will not be sensitive to the herbicides that are present. Similarly, the proteins are regulated by phosphorylation and ubiquitination. So you can modify these sites so that it serves as constitutively phosphorylated form or constitutively dephosphorylated form, or the proteolysis can be minimized if the ubiquitination sites are modified. So all kind of modification is possible to alter the exon, to alter the executing, creating mutation. Knockout is very simple. You create a mutation which causes mutation, which can result in alternate protein sequence and premature termination, and thereby the protein function is lost. You can also, the intron-mediated regulation is also one of the important aspects that can be also regulated. And we can also target the 3' UTR mainly to regulate microRNA binding sites. So you can do all kind of modification in the genome editing, or to create or modify the expression of the gene in its native place. So that's why this is very important tool in any input project to decipher the gene function of the genome wide scale. So we started this work in 2018 with the project that was sponsored by the National Agriculture Science Fund. I must thank Pradhan Mahapatra, then the Director General. So he gave this project because none of us had any experience in the genome editing, only we had some experience in the transgenic development. So ICR believed in us, and with the help of multiple institutes, we started this program. And since then we are running various programs. So one of the outcome of this project only I will be presenting today. So I was always interested in improving the cross variability, stress tolerance. Because that is most important limiting factor in the agriculture product. So this one drought and salt tolerance is the most important stresses. Drought and salt are the most important stresses that prevail in the country. So we were interested in these two stress tolerance from our early career, and this genome editing came as a tool. So this paper which I am showing is a paper which was published in 2009 by Genes and Development by Chinese group of researchers. They screened large number of EMS mutation and they identified a mutant which they named as drought and salt tolerance. And when they did various cloning, they found that it encodes for C2 has to which is a transcription factor that binds to several target genes and it reduces the expression of different target. So they have shown that it is binding to peroxidase 24 precursor and it induces the expression of this peroxidase. So the mutation, single base mutation in this gene was they identified was is the nuclear localization signal. This DNA binding domain, zinc finger domain of this transcription factor. This simple mutation prevents this protein from binding to the promoter of the target genes, thereby it is inhibition of the expression of the target. So when there is a mutation in this gene, it is not able to bind to this precursor, the peroxidase 24 precursor, and thereby it is not able to produce the peroxidase 24. The peroxidase 24 it carbonizes the hydrogen peroxide, which is necessary for stomatal growth. So because the DST is absent, the downstream process of the ABA mediated signaling is faster and thereby induce drought and salt tolerance. Immediately after this paper came, I sent this paper to Master. He was coordinating this project on Reginald. So we were searching for this kind of mutant in this project. So we got some mutant, but we could not get any mutation in the DST field. So even if you know the gene and even if you have a big population of EMS mutation, it is not easy to get mutation of ever in the gene. That's why I want to say this. So you can use various approaches. Some people say why not EMS induce mutation? We do large mutants. So with such a large effort, also we did not get any mutation in the DST. So we started this work in our lab to edit this gene by using the CRISPR-Cas9 editing method. One of my mentor they started working on the genome editing from 2013. So I contacted him. He kindly sent the vector and details how to clone the guide and how to proceed this. So using this the vector which was given to him, we started this work in our lab. We designed two different guide RNAs which are the bold clamp site and the clamp site is and also the cleavage site is marked here. So these guide RNAs are cloned in the vector that we have obtained, and these vectors, these sequences are binary cloned into the P-carrier 1300, which is a binary. This one we used it for genetic transformation. For designing guide RNA, I think the classes will be there. What kind of tool that we use. That time these two are the most common, CRISPR plant and CRISPR. These were very common. While designing guide RNA, it gives all the details like what should be what is the on-target efficiency, what is the off-target efficiency, target efficiency, and then the secondary structure of guide RNA. All these things are taken into account when designing guide RNA. And then we used this vector for transforming the rice cultivar, Mega is cultivar. Fortunately, by that time we had good transmission for transforming the inputs. So we obtained several transgenes. We confirmed by normal PCR method, and then did a PCR amplicon of the target region of the DSTGs and Sanger. So once you get Sanger sequencing, if there is mutation, you can easily identify because you know where gas may be squared to cut the PAM site. It will be cutting about 3 base pair upstream to PAM site. So if you see the various nucleated sequences that you get at the Sanger from the site, you will see all the variation near the PAM site. If there is any variation near the PAM site, which means the double peaks appear from the PAM site, then you know that there is a because you will get one normal copy and edited copy. But sometimes you will get a clear chromatogram also because of the deletion and homozygous mutation in the plant. So this you can also use by using different tools that are available. Earlier we used the SPR-ID and DST. Now there are several other programs that are also. So we created large number of mutants. So these are 90 different combination of mutants that we created, and then we selected the 5 different mutants because each of these mutation they create different values. So you can see that is exactly at the cut place one deleted, that is deleted, or two deleted, that is deleted, or between the two cut sites whatever sequence was there that was completely deleted. In the second guide RNA again either it is a single base insertion or single base deletion or large deletion. So they all produce different kind of proteins. So that's why we selected these kind of mutants for our comparative analysis, and you can see that the kind of proteins that they are different from that of the wild type. So next question is, once you get the mutant homozygous mutant, you observe some phenotype, but always there is a question whether it is because of the on-target mutation or because of the off-target mutation. If possible, you can also search for the off-target mutation in DNA, and you can confirm that is the mutation that created only is the mutation. The other concern was people thought that the genome editing created a lot of off-targets, and that's why off-targets we consider when you are evaluating the genome editing. So this is an example where if you do a conventional background splitting, how many genes you are carrying forward even after times you do that. So still there will be large part of the donor parent will be there. Considering that there is no linkage, otherwise you will have even more number of background genes. When you use conventional mutagenesis also, you get large number of background mutations. This is one of the paper published by where they have sequenced different EMS induced mutation, and you can see how many number of mutations are there in a single mutation. And almost when you compare with the wild type and the mutant, you have more than about 16,450 SNP variation, which means you have so many mutations on a single mutant what you are and also there were large number of mutants. So even if you do selfing also, you can see for each generation you are going to get how many mutations depending upon the genome size. So then we also thought we should characterize our mutant by doing the target amplification of the predicted off-target and see whether you get any mutation or not. This shows the first line shows the normal target loci, whereas these are the other off-targets. So when the off-targets are there, if there is any mismatch within the seed region, that is within the first 8 to 12 nucleotide sequence, DNA prints are on the 3' bed RNA, you don't get mutation. That is what normally we say. So most of our the off-target loci had some or other mismatch in the potential off-target. So then we amplified all this sequence, then we did amplicon sequence, both wild type and the mutants, because the wild type is mutant and we should not compare with the mutant, but whatever genotype that you are editing, you always compare with that genotype. We did not find any mutation in any of the the off-target proteins. So these all help that the existing data and our data, these all help us when we formulated the genome editing to argue that the off-target mutations are not so frequent in the genome editing lines, and also during the breeding program, if there is any negative off-targets are there, that can be segregated. So based on that, guidance that was formulated, the SOPs that was formulated, no off-target data is required if you want to get the exemption for the mutant that you are doing. So this is the phenotype after doing off-target analysis, after doing the homozygous mutant. We compared the phenotype of wild type and the mutant under the osmotic stress and salt stress condition, and these mutants showed better performance than the wild type on both salt stress and osmotic stress condition. Because in the earlier paper it was shown only in the seedling stage, we were interested in trait expressions in the adult plants. So we compared at very high level of salt stress, where we grow normally for any stress treatment of both wild type and the mutant plants are grown in the same part, so that there is no doubt about the stress that you are imposing, whether these plants get equal amount of stress or not. So we can see that the different mutants, they have better ability to tolerate the salt stress, and they produce better grains than the wild type plant. So just to understand to some extent what is the mechanism, we studied the stomatal distribution, and we found that all these mutants they had less stomatal density as compared to that of the wild type plant. This can be also simply experimented by studying the excess leaf water loss. That is, if any ligament is having less stomatal density when compared to its wild type, you can just compare the weight loss after biting the leaf from the plant. If it is losing weight faster, which means it is transferring more loose, it is losing weight less, it means less stomatal density or other mechanism to close its stomach, also retain water. So then we also since they had very less stomatal density, and we want to understand why the stomatal density is less. So which was not known earlier. So we studied all the genes which are in the stomatal patterning genes. So the expression of those genes, if you compare them in the right-hand analysis, we can see that three of the genes they have significantly less expression as compared to wild type plant. The gluten showed less expression as compared to that of the wild type. And then we analyzed its promoter region, and we found that out of the genes they had the DSP binding site, which is responsible for traction in expression. So this validation is required to conclusively prove that the DSP mutant DSP is not able to bind to its promoter or activate the expression of the genes that stomatal number is less. So this paper we published as Martin the NAHEP was the one first to publish any paper on genome editing on plants, like not only at cell level but also at whole plant level. Dr. Siddharth Tiwari has shown that in banana the genome editing can be done. So after that we published our paper in 2020 on genome editing in rice, and this paper is highly cited for so far almost about 231 citations have been received for this paper. We further continued with this. We compared the migration tolerance, that is at the small seedling stage or adult plant stage. This mutant exhibit very high, strong level of stress tolerance. The stress is applied to this plant at higher level. That is, the wild type plant almost they die, but the mutant plant they can do better even in the extreme kind of stress. When the stress level is medium, that is, we are not allowing the plant to to reduce stress which is normally occur in the field conditions. And we can see that all the gluten plant they produce high number of grain per panicle as compared to the wild type plant. And if we compare the grain per plant also, this different kind of alleles they produce higher grain. We also notice that there is always a variation between the alleles in terms of the amount of grain yield and various other phenotypes we observe. So that's why you have to produce multiple alleles and then to our land. Most importantly, the water use is very good, and you can see that the gluten plants they use less water as compared to that of the wild type plant in the controlled condition of RLC, drought stress condition. Not only that yield under stress is important, and yield under normal condition is also important. That at least it should be equal to that of the wild type plant. Otherwise, farmer is not cultivate that area. Even if you say that under stress it performs better, but under normal condition if the yield is less, farmers is not agree because the drought stress is not going to occur. It is highly unpredictable and it is not going to occur every season. It may occur in one season, after that season it may unless otherwise the upland region only we talk. So we examine the yield of this plant under normal condition, and these are what level the yields for the plant was high in different. And also this was due to increase in the Taylor number, increase in the grain number, and increase in the panicle R2 different extent. And in the meantime, we also this gene was cloned as a regulator of the cytokinin oxidase. So we could understand the reason why this mutation creates this kind of mutation, because the DST gene binds to the cytokinin oxidase and it induces the expression. When the DST is mutated, cytokinin oxidase expression is less, so the cytokinin oxidation is promoted, thereby it is promoting the plant. We also compared this in the field condition, and we found that these glutens produce significantly high amount of grain under normal condition, and also under stress condition, in the field stress condition, this can produce higher amount of grain. Especially this gluten, two base perform very well as compared to the 360 base condition. And this are field level yield increase, which is 10 to 21% in this yield increases. So now little bit I will talk about how we are carrying forward this mutation for the field. So this goes with the global regulatory landscape. In many countries, whatever are shown in different colors, and this is different kind of regulation, but in all these countries the genome plants are accepted and as a non-GM. So based on this, earlier at least 4 products were already launched in the market. One is the genome-edited soybean, which is marketed in USA from 2019. So this mutation was created by this Alan approach, and this is a mutation in the start protein which makes this soybean produce high amount of folic acid in order to be polyunsaturated fatty acids. So this will be very useful in very high applications such as frying and other applications, because high amount of polyunsaturated fatty acid, storability and other issues. The other gluten which is commercially available to the consumer is the barbarous, which is again the Japan Institute, they have commercialized this one. This again it came in 2020, and then in 2021 and 2022 the genome-edited fish is already there in the market, whether it is edited or the myostatin derivatives. This gives faster growth or less feed consumption, and it produces the same. So these were already approved by 2020. Here the effort was made from 2018, So initially a committee was constituted for preparing a draft guideline. So I was fortunate to be a member of this committee for preparing the draft guideline. So this guideline was published in 2021, and then for almost 1.5 years there were several discussions. Former Director General Dr. Mahapatra and our Director Dr. K. Singh and ADG Nikki Yadava and they made several efforts to get exemption for the genome edited plant. How to exempt this genome edited plant from GMOs? Because as I told earlier, they are not transgenics and these are distinguishable conventional mutants. So finally, we awaited the office memorandum came from the Ministry of Environment, Forest and Climate Change, because all the GM are regulated under the Environment Action Act and the rules 1989 is that which regulates the and they gave exemption for rules 7, 8, 9 and so on. For these 4 rules it is exempted. That is, once you prove that the SDN1 and SDN2 mutants are free of exogenous introduction, you don't need to get permission for import, export or transport or production or deliberate unintentional release into the environment to cultivate these crops for all these are exempted and this policy is in place. And then once the policy is in place, we also need to have standard operating procedure for regulatory. Otherwise, any of your application go to the regulatory committee, they will ask so many questions because they will have their own doubts, their own expertise. They can ask a number of questions. We did a lot of work under the chairmanship of Professor Ramesh to bring out the standard operating procedure for regulatory review of genome edited plant, where the regulation is very simple. You have to show that the mutant plant that you have generated is not having a selection marker by using a phenotypic selection approach. And second one is by using simple overlapping PCR approach, you have to show that the mutant is not having any of the foreign DNA that you introduced. So for our mutants, we also did the same thing, and you can see that this is the wild type mutant plant, and this is the mutant from which we segregated and obtained the final mutant. So you can see that the transgenic plant, which is from which the final mutant version was obtained, is showing the resistance here and the hygromycin, whereas the final mutant and the wild type is behaving same, which means that they are sensitive, and it shows the absence of the selection marker or its expression. And then we have to prove that this plant is free from introduced exogenous DNA. So for this you need different controls, because you are using PCR method, you also need to have a positive control and negative control. So for this the guidelines suggested that you should use this actin or any of the single copy gene to show that the DNA that you are using is good. Because when some of you, those who have isolated DNA and used it PCR, you might have noticed that sometimes there is no amplification because of the poor quality of DNA. So you have to show that the DNA which you have isolated is of good quality. So for that, use this actin or any housekeeping gene or a single copy gene as an internal control. So with this, we have shown that the template that we are going to use for showing that the mutant is free of exogenous DNA, namely the wild type control, that is the mutant, wild type variety which you used for generating the mutant, and the wild type DNA is spiked with the vector DNA. So this will act as a positive control. So all primer pairs should amplify with this wild type plus spiked DNA template. And the transgenic line, that original transgenic line from which we obtained the final genome edited plant. So this is also again a positive control. And this line, the mutant line and the wild type line, it should be free from the foreign DNA. So we showed that this actin and DSD, they can amplify from this one, and to show that the mutant is free from introduced exogenous DNA, we should do a overlapping PCR, and the PCR is, each PCR amplicon should be of 500 base pair or less than 500 base pair in size, and this amplicon should overlap at least 50 base pair. So that is the guidelines suggest. Based on that, we designed about 49 primer pairs we used for showing that the editor mutant is free from foreign DNA. So this shows the details of what are the primer pairs and what are the amplicon size and what is the kind of overlap that are there between the PCR amplicon. So by using this, we have shown that the mutant plant and the wild is free from the introduced exogenous DNA. So here you obtain in the wild type plus vector spike DNA, you should have band in all the PCR reactions, and in the this transgenic plant from which you obtain your final genome editor line will have band depending upon whether the primer falls in the tDNA region or outside the tDNA region, depending upon the type of insertion that has happened in the original plant. So by this approach, we have shown that the mutant plant that we generated is free from foreign DNA. And then the IBSC, the Institute Biosafety Committee meeting was conducted, and the Institute Biosafety Committee has to accept your data, and they will certify that finally that this mutant is free of introduced exogenous DNA. So once your IBSC is convinced, we have to send all the data to RCGM, and then RCGM finally approves that it is working. After that, IBSC issues certificate. So this mutants, they were exempted on 31st May 2023. After that, these mutants were sent for All India coordinated trial. And besides this mutant, in our NAHEP funded project, the IARR, they have developed the high grain number rice line, which is also under active trial. So these two mutants are under active trial now. NRRA has also developed a high yielding Swarna mutant line, which they have got exemption from their IBSC. Still RCGM approval is pending, and hope that this is also going to come into field testing soon. So already we have a lot of like product in the field, and these are highly recognized. These are already noted by leading journals. So currently we are working with other projects such as the NAHEP funded project on genome editing in different crops: rice, mustard, soybean, chickpea, pigeon pea, and sugarcane. And recently, as told by our Deputy Director General, we are also working on genome editing in different crops. So it is about a 500 crore project. So you have a lot of opportunities, almost about 24 crop science, 25 crop science institute, 10 horticulture institute, 4 institutes of engineering SMT, and 2 institutions from the fisheries SMT, and 14 institute from animal sciences SMT. They are all working on genome editing with this mega funding provided by the government of India. So the potential of genome editing is very well recognized and appropriately funded by the government of India. So it has a lot of potential as a tool to develop product to meet the sustainable development goals and beyond to achieve the ambitious targets. And it is also a functional genomic tool. That is, as I told, whatever modification you want to do, you can do it in its native place. So it is the most efficient functional genomic tool for gene discovery and analysis of gene function. One of the limitations that currently we have, that this technology is patented technology. So you need to have a license if you want to commercialize any of the product developed under, by using genome editing. So currently ICR and the government of India is in talk with the leading institute to get this license. So it is also has like a lot of global market, and you can see that it is very fast growing industry. So finally, I want to conclude that you should not think that you don't have facility, you cannot do. It is the question that gets you the Nobel Prize. After that, you just add lab coats, coffee, and air conditioning. So everything is in your mind. So if you want to do, you can do it from wherever you are coming. So finally, I thank my team, and especially I want to thank Santosh. He has done the DST work that I have presented. I think he will be discussing with you in some of the class. And all my students, my co-PA, Dr. Shivani, Archana, Soham, Panchal, they are all associated with this training program, so you will be hearing. And also my co-PA is from Genetic Division, Gopal, Panchal, and Director. I am thankful to Professor Jenkins. I think because of I was able to start the program, and I am thankful to our former Director General ICR and current Director General ICR for supporting the genome editing work that we do here, and Dr. T.R. Sharma, Secretary Director General Crop Science, under whose leadership all the genome editing work is being done. And he is also chairman of the committee to review all the genome editing work in the country. And Dr. D.K. Yadav, ADG seat, and he was also former ADG NASF for helping us in the project, and also he is the mission director for the current genome editing project. The current ADG NASF, Dr. Jitendra Kumar, and Dr. A.K. Singh, former director IRI, for supporting this work, and also for playing very key role in bringing the genome editing policy in the country. So before concluding, just I want to say that one who solves the problem of water is for 2 Nobel Prizes, one for science and one for peace. So still these 2 Nobel Prizes are waiting for any one of you. So please do get this Nobel Prize and also save the world. Thank you all for the session this week. So after it is a lunch time, if you have some any questions, I am ready to reply.
If you have any questions.
Sir, I wanted to ask regarding the off-target mutation when you said that you compared with the Nippon-Board genome. So while comparing, didn't you face the SNPs which could create mismatch with the variety on which you work?
So I think I told that you have to compare. You should not compare with Nippon-Board. You should compare with the variety of your interest. So we compared with MTU 1010. So we have not compared with the Nippon-Board. So always you have to either when you design guide RNA, you should have the sequence of the gene from the gene of your interest which you are going to work. And also for doing any analysis, off-target analysis, even mutant detection, you have to use the wild type which you use for genetic transformation as the comparator. You should not use the like reference genome that is available.
Any other?
Hello sir. Suddenly I have made slide in which you show the 6 elements and all the functional elements of gene. There one A, P, G, P, G, A, that signals was there. So what it signifies? Probably in third or fourth slide. That's one. ATG and TGA. What is it?
So which one you are saying? ATG is the start codon of translation. So from where the protein translation starts. This I have used to show that like See, when any gene is transcribed, if it is mRNA, this protein coding gene, it has the translation initiation codon, and this is the termination codon. Here, from here, the protein starts, and here when the TGA comes, because the ribosome reads 3 nucleotide to produce one amino acid. So when it sees TGA, it goes up. So this is the place where the protein is produced. This I have put that because in the 5' URL also you will have some ATG. That is before the actual coding sequence starts. In the 5' UTR, in several genes you will have ATG codon. So ATG sequence will be there. The ribosome starts translation from that ATG, and then after few amino acid, there will be like after few nucleotide, there may be a termination codon. Then after that, it falls up. So actually, instead of translating this protein, it will try to translate this small protein. So that lot of energy is wasted in that process. So if you mutate the initial ATG which is there in the 5' UTR, you get more translation from the normal this protein that you expect.
Okay, sir, so is it better to design a guide RNA for 5' UTR rather than exon 1? Is it better approach?
Which one?
To design a guide RNA. Suppose I want to design guide RNA. And I design one guide RNA for exon 1 and also a guide RNA for 5' UTR. So 5' UTR approach will be better.
So 5' UTR, like if you know the this kind of 5' URLs are there, you can design. Otherwise, you can if you want to knock out, you can design primer anywhere to knock out. Maybe exon 1 you can design so that there is no protein is produced. So it depends upon what kind of the stronger mutation you want to produce. You can design in any place exon 1 or exon 2 depending upon the protein function and what are the domains present in the protein. So if you see do a protein domain analysis and then you can decide where you want to create mutation. If you want to protein delete, you can delete from the first exon itself.
Thank you, sir.
In case of rice or model plants, there are lots of gene database available. But recently the need of food security and nutritional security, the world is tend towards millets. So millet database is very unknown for gene specific gene, but millets have lots of potential. So how we incorporate how we retrieve these genes data to incorporating our model plants or staple plants.
So in millets, you have finger millet genome sequence, foxtail millet genome sequence, and this one pearl millet genome sequence. So many genome sequences are available. They may not be available in as good form as the rice genome sequence is there, but these genome sequences that are available. So even if it is the complete genome sequence data is not available, you can always amplify the orthologous gene from the target plant, and still you can design guide RNA and use it. Only thing is that if there is any off-target effect, you will not be able to predict beforehand. But once you create mutant, you can just screen the mutant. If any plant is with undesirable character, you can just omit it. So now, because the off-target mutations are not part of regulatory process, you need not generate any data on the off-target mutation. So you do mutation only with the target gene. You can just sequence your target gene from the target plant, design guide RNA and develop mutant. After that, you screen the mutant by phenotypically. And if the mutant plants are giving only the target phenotype, the adverse effects are not observed, then you can use that mutant for commercialization. So it is not compulsory that you have to show all that.
So while designing SGRNA, the polyploidy nature of crop or species matters or not?
It is advantage also, it is also disadvantage. I think Naveen Bist will make one presentation. So mustard is an allotetraploid. He designed guide to target all its formulas that are there, as well as different family members of the target genes. So with the single guide RNA we can edit multiple genes. Otherwise, if the homology is very high and you want only mutation in only one of the 2 or 3 homologs, you make mutation, and because in the first time when you make mutation, it is not under the control, unless otherwise you can design a specific guide RNA, which is often very difficult. So you can create mutation, and then after crossing you can segregate the mutation in the different genomes. That is also possible.
So I think there may be many more questions, but let's stop here because this is the first lecture. So as we progress in the training, a lot of doubts will be cleared. So thank you very much, sir, for excellent talk and setting the tone of the conference training. Now I request Dr. Pradeep Jain, sir, to please felicitate our first speaker, Dr. Vishnu.
Privilege to present this Dr. Vishwanathan, and one of the most illustrious scientists of the country, I would say. Our own Manuel Charpentier or Jennifer Doudna, I would say.
So sometimes if I get time in the evening hours, I will come and interact with all of you.
Bus is at the front of the building. We just have to go to Sindhu Guest House or lunch and then come back.
Over.